Hi, Assalamualaikum warahmatullahi wabarakatuh, and a very good day. I am Madam Nur Shamim, and today we are going to learn on a new Chapter 7, Plant Tissues. Let's begin. In this topic, you will learn two objectives. The first one, you must be able to state and define terms in tissues. The second, you must be able to identify, to draw, to label, and to describe all the characteristics of the plant tissues. In this chapter, you will also learn on the anatomy and physiology part of plant. These include the meristem tissue, the vascular tissue, the ground tissue, and the dermal tissue. Okay, let's catch up on previous lesson. So this is the structural organization in organism. It starts with the molecule, organelle, later it will form cells, and the cells will form the tissues, and the tissues will form an organ, and later they will form a community. Tissue system consists of dermal, vascular, and ground. For dermal tissue, it is the outer protective covering like our skin. It forms the first line of defense against physical damage and pathogenic organisms. For non-woody plants, the dermal tissue usually consists of a single layer of tightly packed cells called the epidermis. For woody plants, the protective tissue known as periderm. Let's see for the second tissue, vascular tissue, which carry out long-distance transport of material between the roots and shoots. The two vascular tissues are xylem and phloem. Xylem conveys water and dissolved minerals upward from root into the shoots. Meanwhile, phloem, it transports organic nutrients such as sugar from where they are made of, which usually the leaf, to where they are needed, especially the roots and the site of growth like developing leaf and fruits. Okay, tissues that are neither dermal nor vascular are part of the ground tissue system. It includes various cells specialized for functions such as storage, photosynthesis, metabolism, and support. There are three examples of ground tissue cells: parenchyma cells, collenchyma cells, and sclerenchyma cells. Later, we will discuss the function of each cell. So far, we have learned on the structure and function of tissues in plant mature organs. But how this organization arise? Let's see the next slide. The major difference between plants and most of animals is that plants can grow at any given time, but animals' growth is limited to embryonic and juvenile period. What makes plant can grow like this is due to the meristem tissue. There are two types of meristem, apical meristem and lateral meristem. Apical meristem located at the tips of roots and in the buds of shoots. It provides additional cells that enable the plant to grow in length. This process is known as primary growth. So this primary growth will allow the root to extend throughout the soil and shoots to increase exposure to light and carbon dioxide. For addition, the root tip is covered by a root cap which protects the delicate apical meristem as the root will push throughout the harsh soil during primary growth. Later, I will show you the structure and the location of the root tip and the root cap. The apical meristem will form the protoderm, ground meristem, and the procambium. This protoderm will later specialize into epidermis. The ground meristem later will form the parenchyma, collenchyma, and sclerenchyma tissues. Meanwhile, the procambium later will form the primary xylem and primary phloem. The image below shows the primary growth happen in a shoot and in a root. Let's see the right picture. The protoderm, the ground meristem, and the procambium is the primary meristem that later will specialize into certain cells. There are a root cap at below. This root cap is functioned to protect the apical meristem, to protect the root apical meristem when the root pushes through the harsh soil during the primary growth. In this slide, it explains the apical and lateral meristem. If you can see the apical meristem, which involves the terminal bud, which contain a shoot apical meristem. Okay, for the lateral meristem, it involves the cork cambium and the vascular cambium. So in apical meristem, it involved with the primary growth and produce increase in length. Meanwhile, the lateral meristem, it involved with the secondary growth and cause the plant to increase in diameter or thickness. This image shows the leaf primordium, which leaf will arise from, and the apical meristem of shoot, and the axillary bud primordium, which later will form the lateral shoot of plant, the ground meristem, which later will form three types of tissues, the parenchyma, collenchyma, and the sclerenchyma tissues, the procambium, that later will turn into primary xylem and primary phloem, and the protoderm will form into epidermis and periderm. This image refers to the primary growth of roots. The primary growth of roots involved with the mitosis process, which concentrated in the zone of cell division. So, this apical meristem will focus or concentrated in the zone of cell division. The apical meristem will also maintain the root cap by generating new cells that replace those that are sloughed off. Remember, this root cap is functioned to protect the apical meristem when the root try to pushes the harsh soil during primary growth. And later the cells become functional mature in the zones of maturation. Lateral meristem involved in plant secondary growth. Plants use lateral meristem to grow in diameter or thickness, and there are two types of meristematic tissue in lateral meristem. The first one is vascular cambium, the second is cork cambium. Vascular cambium will form the secondary xylem and secondary phloem. Meanwhile, the cork cambium replace the epidermis with periderm, which is thicker and tougher. This is another example of plant secondary growth. Let's see for the year 1. Normally it goes into primary growth, and for the year 1 late summer and year 2 late summer, the plant is increased in their diameter. So it means the plant is undergo the secondary growth. For herbaceous or non-woody plants, the primary plant body is usually the primary plant. So that is for herbaceous plant. Meanwhile, for non-woody plants, the primary growth It consists of youngest part which have not yet become woody. The plant stem and root will start to grow in girth. Girth means to increase in diameter, and this is called the secondary growth. You must remember, the secondary growth is caused by the activity of lateral meristem. This is the secondary growth in root. Lateral meristem consists of cord cambium and vascular cambium. And here there are root hairs, which are the specialized of epidermis. And we have ground meristem here, which later will form into three types of cells. The procambium, which later will form into primary xylem and primary phloem. The apical meristem, where the zone of cell division is concentrated here. The root cap. The root cap is functioned to protect the apical meristem. From this, I hope you can be able to differentiate the apical meristem, which involve with primary growth, meanwhile lateral meristem involved with secondary growth. Let's catch up. Vascular plant consists of root system and shoot system. The root system will anchor plants and penetrate soil to absorb water and ions. Meanwhile, the shoot system consists of the stem, the leaf, and the vegetative shoot. This is a plant body diagram. Plant consists of shoot and root system. From this diagram, you may look up which part that involve with the primary growth and which part involved with the secondary growth. Again, we revise on the previous lesson. Plant tissues consist of three types of tissues: the dermal tissues, the vascular tissues, and the ground tissues. Please remember the function of each tissues and their role in plant growth. Now let's start with the first tissue, dermal tissues. If you still remember, dermal tissue will form into epidermis in non-woody plants and periderm in woody plants. These epidermis, they have specialized characteristics in each organ. For example, epidermis will form or will specialize into gut cells in leaf and stem that is used to control the gas exchange. Besides, these epidermis also will specialize into leaf trichomes. Leaf trichomes is the outgrowth of epidermis. Some of the aromatic leaf, they have trichomes that can produce oils such as mint leaf. This oil is functioned to protect the plant from herbivores and diseases. And last, this dermal tissue also can specialize into root hairs. Root hairs are very important for absorption of water and mineral, and it is located near to the root tips. These are the examples of specialized cells from epidermis. The gut cells, which control the opening of the stoma, the trichomes, which is the outgrowth of leaf, and the root hairs, which involve in the absorption of water and mineral in plants. The second is ground tissue, which consists of parenchyma cell, collenchyma cell, and sclerenchyma cell. Mature parenchyma cells have primary walls that are very thin and flexible. This cell can perform most of the metabolic functions of the plant, synthesizing and storing various organic products. For example, photosynthesis occurs within the chloroplast of parenchyma cells in the leaf. and the fleshy tissue of roots mainly composed of parenchyma cell. For collenchyma cells, they are normally found or grouped in strands or cylinders. It is functioned to support the young parts of the plant shoot, and this collenchyma they has thicker walls compared to parenchyma cell, but the cell is lack of secondary wall and absence of lignin. Therefore, they provide flexible support without inhibiting the plant growth. And the last is sclerenchyma cell. These cells are more rigid compared to collenchyma cell. It is also functioning as supporting elements, but it has very thick secondary wall and strengthened by lignin. Mature sclerenchyma cells cannot elongate, and normally these cells are found at the part which have already stopped growing. In other words, these cells are dead at functional maturity. That's why there is no growing at all for sclerenchyma cells. There are two types of sclerenchyma cells, sclerid and fibrous, and both of them are entirely specialized for support and strengthening. Sclerid is shorter than fibrous, irregular in shape, had very thick lignified secondary walls. Meanwhile, the fibers are usually arranged in threads, long, slender, and tapered. Later, you will see the structure of these two. This table refers to the differences between parenchyma cells, collenchyma cells, and sclerenchyma cells. You may read their differences and make comparison. This is the ground tissue diagram, which consists of parenchyma tissue, collenchyma tissue, and sclerenchyma tissue. From this diagram, you may differentiate the structure of each ground tissue, the presence of the lignin, which related to its supporting functions, and the thickness of each cell. Here is the vascular tissue, which is the last tissue system in plant. There are two types of water-conducting cells in xylem. They are tracheids and vessel element. They are formed in the tubular and that at functional maturity. Let's see the structure of tracheid. The tracheid are long, thin cells with tapered end. The water moves from cell to cell through the pits. The secondary walls of tracheid are hardened with lignin. This will prevent it collapse under the tension of water and also provide support to the xylem structure. For the vessel element, it is much wider and shorter, thinner wall and less tapered compared to tracheid. These vessel elements are aligned end to end, forming long micropipes, and they are known as vessel. The end wall of vessel element have perforated or hole. This hole will enable the water to flow freely into the vessels. Unlike the water-conducting cells of the xylem, phloem are alive at functional maturity. The sugars and other organic nutrients are transported through sieve tubes, which consist of chains of cells that call sieve tube members. Even though phloem is alive, it lacks some of organelle such as nucleus, ribosome, and vacuole in order to make sure the nutrient can pass easily through the cell. The end walls of sieve tube members, called the sieve plate, it have pores. These pores will facilitate the flow of fluid from cell to cell. Together with sieve tube members or alongside is a non-conducting cell called the companion cell. So this companion cell is located beside the sieve tube members. The companion cells connected to sieve tube members by the plasmodesmata. Diagram shows the xylem and phloem. Let's see the xylem. There is no end walls between the cells, and the water and minerals are conducted one way only. The outer cells are not living. That's why xylem is dead at maturity. For phloem, the end walls, which consists of the sieve plate, they have pores. These pores will facilitate the movement of minerals and sugars. And phloem do two-way movement. The cells are living but need support. Remember the companion cell which located alongside the phloem. The companion cell will give support to the sieve tube members. That's the end of our lecture. I hope you can understand and do revision on this topic. Thank you for your time and thank you for listening to my lecture. Bye.